Welcome back, class, to our third lesson and talking about RNA and protein synthesis. Today we're going to talk about gene regulation, gene expression, and mutation. So buckle up. It'll be a little bit longer of a lesson, but we've got a lot to cover. So we'll get started. You have a couple of learning targets today. By the end of this video, you should be able to describe gene regulation in prokaryotes. You should be able to explain how most eukaryotic genes are regulated. You should be able to relate gene regulation to development in multicellular organisms, describe how mutations change genetic information, and explain how mutations affect genes. So we've got a lot to cover. Our big idea, remember, is information and heredity. And the unit question we've been trying to answer is, how does information flow from the cell nucleus to direct the synthesis of proteins in the cytoplasm? And this is the last bit of that. Today we'll be answering the question, how do cells regulate gene expression? We can't turn all of our well, we could. We don't want to turn all of our genes on all at once. So how do we know when to turn them on and when to turn them off? All right, so let's start with an example of gene regulation. It wouldn't make sense for your body to turn on all your genes all at the same time. And this is because you don't necessarily need every gene in your body on all at once. When you are a baby, you need some genes, and when you're an adult, you need other genes. And so how can our bodies regulate this? And we'll approach this from an example. E. coli is a bacteria, and it's what we'll use as our example of how gene expression can be regulated. So first, some background information. An operon is a group of genes that operates together. Operons can help us regulate genes. Now, I want to make a point that this is an example of prokaryotic gene regulation. Eukaryotic gene regulation is a little bit different. So we're talking about a simple organism, bacteria, E. coli, and its way of operating or regulating gene expression. In E. coli, these genes must be turned on so the bacteria can use lactose as food. So E. coli bacteria has the ability to turn on and off the gene that allows it to break down lactose, which is a type of milk sugar. You can think about this in terms of humans too. Humans can be lactose intolerant, which means they can't have milk, which means they can't have the milk sugar. This is because their body lacks the enzyme to break it down. Where E. coli bacteria has the ability to turn on and off the regulation of the expression of the gene that allows them to break down lactose. So, in E. coli, genes can be turned on so the bacteria can use lactose as food. Or these genes can be turned off by regulatory proteins that bind to DNA and block transcription if they don't need to break down lactose. It'd be like, you don't drink milk with every meal, so you don't constantly need to be making lactase all the time. Lactase is the enzyme that breaks down lactose. You only need lactase when you consume milk or lactose or any sort of milk-based sugar. So, therefore, in this example, we call it the lac operon. So just to recap, an operon is a group of genes that operate together. E. coli have a group of genes called the lac operon. They can be on when the bacteria is using lactose as a food, or they can be off when the bacteria is using something else as a food, such as glucose, which is a non-milk sugar. So, how are the lac genes turned on and off, and why do they even really matter? Lac genes are turned off by these things called repressors and turned on by the presence of lactose. And we'll talk about and identify these repressors in just a second. So here's what it might look like, and I'll walk you through this kind of step by step. So we have these things called promoters, which is where RNA polymerase is going to bind, and then we have operators, which is where repressors can bind. So located in front of the operon's three genes are two regulatory regions called a promoter and an operator. The promoter is abbreviated P, and this is where RNA polymerase is going to bind in order to transcribe and translate these genes to make a protein that will allow the body to break down lactose. And then we have the operator, which is where something would bind to repress the ability for RNA polymerase to actually carry this out, okay? So we've got, and then there's the lac repressor gene, which codes for the repressor protein. These are the lac genes upstream of the operator, the promoter, and the repressor. So I'm going to break this down a little bit more. So if this is our stretch, right, of DNA or of mRNA, right, there's the stretch. And these are the genes that code for lactase. Let's just say, for example, whatever, the substance, the enzyme that's going to break down lactose. We can allow the transcription of these genes to take place. If we allow the transcription of these lac genes to take place, the E. coli will produce the enzyme and break down the lactose and function properly. If there's no lactose present, we're going to block the transcription of the lac genes, and therefore lactose won't get broken down. Okay, so when the lac repressor binds to the O or operator region, RNA polymerase cannot reach the lac genes to begin transcription. If the lac genes cannot be transcribed, then it cannot make the enzyme, which is a protein that breaks down lactose in the E. coli bacteria. So here's where our repressor protein is, and we'll talk more about that repressor protein, so keep your eye on it. When lactose is added to the medium, it diffuses into the cell and attaches to the lac repressor. So that's happening down here. Here is a lactose molecule. It can bond to here. It changes the shape, and since it changes shape, it can no longer bond to the operator. Since it is no longer bound to the operator, it cannot suppress RNA polymerase. Therefore, RNA polymerase can carry out transcription, making the special enzyme that is going to break down lactose. Oops, sorry about that. Okay. Many genes are regulated by repressor proteins. Some genes use proteins that speed up transcription, that is, some genes make it faster, and sometimes regulation occurs at the level of protein synthesis, which means we can either turn off, stop, speed up, slow down protein synthesis, or the making of proteins. Now that is what happens primarily in prokaryotes. It's a pretty easy system. If something is present, then it bonds to the repressor doesn't bond to the operator, and transcription takes place. If something is not present, the repressor bonds to the operator, RNA polymerase can't transcribe the genes. It's as easy as that. Eukaryotic gene regulation is a little bit more complex. They follow some of the same systems, right? But you are a more complex organism, therefore the process is in general, by nature, more complicated. Most eukaryotic genes are controlled individually and have regulatory sequences that are much more complex than those of the lac operon. One of those is what we call a tatabox. So we'll talk about it in a little bit. Here is what we can say is another sequence of genes right here. We've got an enhancer right here, promoter sequences, recognize promoter again, a tatabox, which we'll talk about, and introns and ectrons. Remember that introns get cut out in that mRNA processing before they become mRNA. Operons are generally not found in eukaryotes. So if you look at this image here, notice that operons are not present. We do have a promoter sequence, but we don't have an operon. Instead, we have that thing called a tatabox, and we'll talk about that in just a second. All right, so a typical eukaryotic gene has what is known as a tatabox. This is usually a regulatory sequence of TA, or TATAAA. It just kind of depends. There's the tatabox located there. Factors can either bind to the enhancer regions or bind to the tatabox in order to control gene regulation in eukaryotes. So it would look something like this. You'd have transcription factors that would bind to the enhancers, as noticed here, and then you'd have transcription factors that would bind to the tatabox, as noticed there. This causes the DNA to fold and pushes the RNA polymerase in the correct direction of transcription. If transcription factors aren't present, transcription may not occur. The TATA box seems to help position RNA polymerase in the correct spot. Now, we're still learning about the TATA box, but it is relatively common in eukaryotes, and it has something to do with transcription. Eukaryotic promoters are usually found just before the TATA box and consist of short DNA sequences that basically signal bind here to those transcription factors. By binding DNA sequences in the regulatory regions of eukaryotic genes, transcription factors can control the expression of those genes, similar to that of the prokaryotes and their promoters and operators. Genes are regulated in a variety of ways by these things called enhancer sequences. A lot of proteins can bind to different enhancer sequences. Some DNA binding proteins enhance transcription by either opening up tightly packed chromatin or by helping to attract DNA polymerase or by blocking access to genes. And so some of these, if we go back here, notice the enhancer regions located here and here. Those are places where transcription factors can bind. And if those transcription factors bind there, they can either allow, remember, chromatin is closely packed, which prevents the copying of genetic material. So if we bind to an enhancer region, we can allow that chromatin to relax, which allows the transcription to actually take place. We can help attract RNA polymerase, which notice from our previous image, RNA polymerase, it was what got folded around and pushed forward. Or it can block access to a gene, which could prevent transcription from taking place to begin with. This is important when it comes to things like development and differentiation. Your body needs to be able to turn things on and off in order for you to develop properly. As cells grow and divide, they undergo differentiation, which we learned about a few units ago. Remember when we were talking about stem cells and we did our planarian activity? As cells grow and divide, they undergo differentiation, meaning they become specialized in structure and in function. Hox genes control the differentiation of cells and tissues in the embryo. We'll talk a little bit more about Hox genes in just a minute. Careful control of expression in Hox genes is essential for normal development. Most of these genes are turned on in an embryo to allow arms to develop in the right place, legs to develop in the right place, and other organs to develop where they're supposed to go. All Hox genes are descendant from genes of common ancestors. So here's what we call them homeotic genes. Our homeotic genes regulate organ development. So if you take a look, now we've got the various chromosomes down here. That's what's represented by these chromosomes. We are looking at fruit fly chromosomes and mouse chromosomes to see the relationship and the similarities between eukaryotic organisms. So each of these little colored bands indicates the location of a specific gene. And so you can see some of these genes are conserved. Now, they're not all identical. There is some variation in them, but you can see some of the similarities. Now, those homeotic genes help code for these homeobox genes, which code for transcription factors. So these homeotic genes are collectively referred to homeobox genes that code for these transcription factors. These transcription factors help indicate where and when and what each of these is going to become. And so you can see by the color, the association. For example, the purple band is going to be something down here like this. And in that respect, it'll be like the nose or the mouth or the face area. You can see the development of, I'm trying to find one that's easier to see, this green band here. You'll notice it here, and then you can see the pink and the green bands here. This is going to be the head-neck region located here on the mouse. Then and the yellow one's another one to point out here and here and here. We can see this is part of the limb region, right? And so you can see some of the development taking place. So these Hox genes determine the identities of each body segment. These are present in humans too. If you follow these gene patterns, they're the reason why your head's on your head and your eyes are where your eyes at and your arms where your arms at. Biologist by the name Edward B. Lewis did a lot of research with these. And we can actually, he did research to where he rearranged these genes, and he found that if you turned on or off these genes in certain patterns, you could get organs to grow in different places. For example, you could get legs to grow where fruit fly antennae grow, and you could get all sorts of weird combinations. So the regulation of these homeobox genes or these Hox genes are extremely important to the development of eukaryotic organisms. Now, Hox genes aren't the only thing that play a role. There's a lot of different things that play a role in gene regulation. So of course we regulate genetics on the genetic level, right, with those promoters, operators, Hox genes. But there are other things that can play a role in eukaryotic gene regulation and expression as well. Epigenetics is defined as a change in gene expression without actually changing the gene itself. So on a molecular level, the genetic code is the same, but the expression of that code has been modified. And this can be seen in things like identical twins, right? The genetic code is the same, but the expression of these genes is a little bit different. So epigenetic mechanisms control which genes are on and which genes are off at each stage of, for example, an insect's life cycle, which is why many bugs look alike, but there's also a lot of differences in them. We also have things like environmental influences that can affect the expression of genes. Environmental factors can regulate gene expression, such as temperature. We talked in one of our previous units about how the season in which a butterfly is born can change the color of its wings. If it's born in the colder seasons, its wings are going to be darker to absorb heat so that it can fly. And if it's born in some of those hotter seasons, its wings will be lighter because it doesn't need to be have that dark wing color in order to fly. This can be seen at bunnies as well. Here is a rabbit that is raised at temperatures below 25 degrees Celsius, and here is a rabbit that is raised above 30 degrees Celsius. This is also super common in lizards and sea turtles. It can impact the gender of certain organisms. So environments can also play a role in how genes are expressed. Malayan rabbits, the gene for black ears and black paws and a black tail and a black nose is not fully activated at temperatures above 30 degrees. In order for these genes to be activated, it has to be cooler than 25 degrees. But what are some of the other things that impacts the regulation and expression of genes? What about mutations? So mutations also impact how genes are expressed. There are a couple of different mutations that we'll talk about. Mutations are heritable changes in genetic information, which means if you have a mutation in your body, you could potentially pass that mutation on to your offspring. Mutations fall into two basic categories: gene mutations and chromosomal mutations. We'll talk about the difference between them in just a minute. There are multiple different types of gene mutations. One of them is called a point mutation. Point mutation is the change in a single point or a single nucleotide, and there are three different types of point mutations. You have substitutions, insertions, or deletions. A substitution, as can be seen here, is where you take one nucleotide and you swap it for another nucleotide. A change in the nucleotide sequence can completely change which amino acid is present. So here is the original genetic sequence before a mutation. If a substitution occurs, we're going to completely change this arginine amino acid to this amino acid down here. We also have an insertion. This is where you could just add an amino acid, a nucleotide. If you simply add a nucleotide, you shift all of these nucleotides over, which means you change all of these amino acids completely. Similar to an insertion is a deletion. Actually, it's the exact opposite of an insertion, where you completely delete or eliminate an entire base or an entire nucleotide. Deleting an entire nucleotide is going to completely change the sequence of the amino acid. So here is the original protein, and here are the possible variations of the protein based on different mutations that can occur. Most mutations are silent, which means they don't actually do anything, because remember, you have introns in your body that get cut out. Many mutations are harmful, and a few mutations every once in a while can be beneficial, but we'll talk about that in a second. In a substitution, one base is changed into a different base, as we talked about. Here you can see the original mutation, original sequence, which is totally normal. It gives us CGU. which gives us the amino acid arginine. Now, if we have a substitution take place, it's going to swap out the C for T. This gives us a completely new amino acid, in this case, histidine. Insertion mutations are when a single extra base is added into the genetic code, and a deletion is when a single base is removed from the code, completely shifting the amino acid sequence. Now, when it comes to chromosomal mutations, they're a little bit more complex, and they can be a lot more damaging than some of those point mutations or genetic mutations. Now, some of them can be the same. You can still have chromosomal deletions, duplications, but you can also have things like inversions and translocations. A deletion is where you completely delete a segment of a chromosome. A duplication is where you replicate a segment of a chromosome. An inversion is where you swap the gene's location on the chromosome. And a translocation is when you take a gene from one chromosome and you put it on a totally different chromosome. If you think about the fact that you only have 46 chromosomes, if you start mutating your only 46 chromosomes, you're going to suffer probably some more consequences than if you mutate one base pair in the hundreds of thousands of millions of billions of nucleotides that you have located in your DNA. So a deletion, there you've completely deleted the B segment of your chromosome. With a duplication, we've replicated the B segment of the chromosome. With an inversion, we've swapped the B and the C segments. And in translocation, we've completely removed one section of a chromosome and put it into a new chromosome. So mutations can be harmful, they can be beneficial, or they can have no effect on you at all, as we've kind of talked about. Some mutations arise from these things like mutagens, and these can be chemicals or physical agents in the environment. For example, skin cancer. Skin cancer oftentimes comes from the fact that you're out catching some UV rays. UV rays are a type of mutagen. As your body absorbs those UV rays, it can cause changes in your genetic code. The changes in this genetic code can cause your cells to grow uncontrollably. Remember that the cell cycle has these checkpoints. If you cause a mutation in your genetic code, it can turn off one of these checkpoints. And if it turns off one of these checkpoints, it can cause your cells to grow uncontrollably, which could lead to cancer. Many cancers actually come from mutagens or mutations in the genetic code. Where's this genetic code? In the cell cycle, the stop and start portion of the cell cycle, because everything in your body is regulated by the genetic code. Most of the effects of mutations tend to be harmful, although a lot of them also silent. Some of the most harmful mutations are those that drastically change the protein structure or gene activity. For example, the turning off or on of a portion of the cell cycle. Another example is sickle cell anemia or sickle cell disease. Sickle cell disease comes from a mutation in the genetic code that changes the shape of your red blood cells. A normal red blood cell has this round shape right here, as you can kind of see in the picture, and a sickle cell has this shape right here. In sickle cell disease or in sickle cell anemia, what happens is that most of the individual cells have this sickle shaped. Now, why does this matter? Your red blood cells are shaped like a donut, specifically so they can carry oxygen throughout your body. If they have this sickle shape, they can't carry oxygen as efficiently, which can make it harder for you to breathe and harder for you to regulate oxygen throughout your body. Now, many people with sickle cell anemia or sickle cell disease can live full lives. They have to take medication. But you can also be a heterozygote for this disease and have some benefits, because sickle cell disease is also said, the sickling shape of the cell is said to protect people from malaria. Mutations oftentimes produce proteins with new or altered functions that can be useful to organisms in different or changing environments. One example is being a homozygote for sickle cell disease, which means you have both the dominant, which means you have full-blown sickle cell anemia or sickle cell disease, is harmful. If you're a heterozygote, basically what it means is that some of your cells are sickled and some of your cells aren't sickled. Sickled cells are less likely to get malaria. So if you live in an environment that's heavy with malaria, it's beneficial for you to have some of your cells be sickled. Now, again, mutations change the genetic code in your body. Because they change the genetic code in your body, they change the proteins in your body that can be made. The most harmful mutations are those where the protein is completely changed and potentially damaged. And now the protein has new or altered functions, which could be harmful or beneficial depending on the environment you live in. So, can you tell me how cells regulate gene expression? Do so as specifically as possible. And if you can't, there's some great information in your textbook to help remind you. Additionally, ask yourself, can you describe gene regulation in prokaryotes? Can you explain how most eukaryotic genes are regulated? Can you relate gene regulation to development in multicellular organisms? Can you describe how mutations change genetic information? And can you explain how mutations affect genes? There's a lot of information in this lesson today. So feel free to go back, reach out, ask questions if you have them. And as always, I will see you next week.